Hello, I am Biva Patel and I will be covering gene regulatory network. We will be first covering gene regulation in prokaryotes, then in eukaryotes, and then gene regulation using recurrent neural networks with random forest algorithm. DNA is used to give instructions for production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which will be turned off. This process is called gene regulation. Now we will be seeing gene regulation in prokaryotes. There are many ways in which genes are regulated. In your human body cells, you can have proteins that can bind to a certain gene regions to increase the rate of transcription of the transcription enzyme RNA polymerase. This is positive transcription. Or you can have proteins decrease transcription to a point that they may not be transcribed at all. This is negative transcription. Now, let's meet the key players of the gene regulation. That is RNA polymerase, promoter and operator. First is the RNA polymerase. It is a builder enzyme. The RNA polymerase is needed to start the transcription. The thing about RNA polymerase though is that transcription gets a little confusing for RNA polymerase without somewhere to bind. Well, for this reason, RNA polymerase needs a promoter. A promoter is a sequence of DNA where RNA polymerase can bind. So you think that that's it. You get RNA polymerase attached to a promoter and boom, you can make your mRNA, which eventually will be used to make protein, right? But it doesn't happen so. There is a sequence of DNA called an operator. The operator is a part of the DNA where a repressor can bind. If bound to the operator, the repressor blocks the RNA polymerase and hence the RNA polymerase cannot move forward and hence no mRNA can be made. For example, here you can see that the RNA polymerase is blocked by the repressor, hence it cannot make mRNA. But for situations like that in lactoperon, the lactose molecule binds itself to the repressor. This changes the structural properties of the repressor. Hence, it cannot bind to the operator anymore. This clears the path of the RNA polymerase and hence it is now free to make the mRNA. Now, we will be seeing gene regulation in eukaryotes. Gene regulation in eukaryotes is a little different than that from the prokaryotes. Here, we will use what are called as transcription factors. So, the RNA polymerase is here, but RNA polymerase cannot get on until we have a number of transcription factors present. And so, let's say we want to make a protein. To do so, we have regulatory factors. Those are actually made by the regulatory genes that could be somewhere else in the DNA. First of all, the transcription factors will allow the attachment of RNA polymerase. We will have other transcription factors that will actually hold it in place. And so, in order to make the gene, the DNA upstream will actually have to fold. It will get more transcription factors and you will notice that there is nothing still going on. We still don't have transcription. And for it to actually happen, the DNA actually folds back and activates that RNA polymerase. And so now it can actually make the protein or RNA that eventually will make the protein. This is how gene regulation actually occurs in eukaryotes. Now we will see how gene regulatory networks are formed from these gene regulations. Gene regulatory networks is a accumulation of molecular regulators that interplay among themselves and with other materials in the cell to control the gene expression levels of mRNA and the proteins. The regulator can be DNA, RNA, protein, and a complexes of these. It can be through transcribed RNA or through a translated protein. Each mRNA molecule goes on to make a particular protein, and sometimes this protein will be incorporated at the cell covering or within the cell to give it a particular design or properties. In other cases, the protein will be an enzyme. Some proteins trigger other genes, and these are transcription factors that may play the main role in the regulatory networks. The gene regulatory network is modeled as a directed graph. G is equals to VE, where V represents the set of all nodes or genes and E is the set of all edges. An edge E in the set E represents physical or the regulatory interactions between the two genes. Genes can be viewed as nodes in a network, with input being proteins such as transcription factors and output being the level of gene expression. The node itself can be viewed as a function which can be obtained by combining basic functions with inputs. Thus, by modeling the gene regulatory network, we can get a weight matrix giving us the output of interactions between the two nodes. Now, using this output weight matrix value, we can calculate the value of gene expression required for a particular change in the cell. In our project, we have coded a gene regulatory network of an yeast using recurrent neural network with random forest algorithm. Source code. Here you can see the gene regulatory network.py. This file contains our main source code. The README file contains the instructions and the requirements needed for the file. .mat is our microarray dataset which contains 6178 genes and its corresponding 77 timestamps. And yeastnames.mat is our dataset which contains the names of the corresponding 6178 genes. _grn.csv is our output file which contains a matrix of 6178 cross 6178 genes. In order to run the gene regulatory network.py file, you go in the terminal, type python gene regulatory network.py and then enter. This runs our Python file. We are going to be using recurrent neural networks using random forest algorithm for this code, and we are going to be using two datasets for this code. One is yeastnames.mat and the second is yeast.mat. Yeast.mat contains 6178 total genes and its corresponding mRNA values at different timestamps. And yeastnames.mat contains the names of those same corresponding genes. Now these values, that of yeastnames.mat is saved in data_yeast names and that of yeast.mat is saved in data_yeast_features. The data frame allows us to create a multi-dimensional matrix, allowing to take combinations of rows and columns from different matrices. Here, we are accessing the value of dictionary key called YYSTR, which is necessarily a 1D array. In yeast features, we are accessing the key value yeast, and which consists of the different features of yeast. And in data initial, we are creating a matrix with features of genes as columns and their names as rows. Now we are storing the value of the data initial to temp, and here in number genes we are taking the total number of keys that are, or the total different types of genes that are present, and here the number is 6178. In network, we are creating a matrix of 6178 cross 6178 length. 6178 is actually the number of yeast genes present in the dataset. In this for loop, we will see that all the genes, that is 6178 genes, one by one will become the target gene. Here, all the genes are put in the key list, that is 6178 in number. Then one of the gene will become a target gene, and that gene will be removed from the keys list. Now, these keys now become the input genes. Now we see that the matrix X contains the list of the input genes and the matrix Y becomes the target gene. Hence, now the training of the recurrent neural network starts. Here we are initializing the random forest regression object, and n underscore estimators is equals to 200 means that we will be creating 200 decision trees in the forest. And here it is creating 200 trees using x matrix as input and y matrix as output matrix. Mean squared error is used to compute the weights between the target genes and the input genes. This helps us know how these genes are related to one another. This data is stored in the features importance matrix, which is 6178 cross 6178 in number, and this contains different weights between each and every gene. In this for loop, you can see that the features importance matrix is stored in the network matrix. This is then stored into our output file, that is our east_grn.csv file. This is the file which contains the 6178 cross 6178 matrix. Okay, so now let us understand how random forest regression works. Okay, so what is regression? Regression is a data mining task of predicting the value of target by building a model based on one or more predictors. Here our predictors are going to be the decision trees. In random forest, decision trees are used as the predictors. For example, now we can see that let x and y be the input values, that is x is the input and y is the target gene, and from the set of inputs from the data set, the algorithm splits the x-axis in one or more halves. That is, for example, here you can see that it has divided the x-axis in 4 different regions by 3 different values. These values are taken randomly, and here the values are x1, x2 and x3. From this, the following decision tree can be generated. Here the decision here you can see that x is our input, and x, after undergoing a series of decisions, that is in yes and no form, it will land up in either A, B, C or D. Here you can see that x and x3 and x2 are our decision nodes, and A, B, C, D, our leaf nodes represent the decision of the numerical target, and the root node is called the best predictor node. After this process, A, B, C and D, that is the leaf nodes, will contain a series of output values, that is the Y. A, B, C and D each will have a different mean of their own, and all these values, all the different values of Y their mean square error will be calculated with their corresponding Y cap. For the values of Y for which the mean square error is the least, that value is the optimal value. We are having 200 such decision trees. Hence, each input and target gene pair undergoes 200 such decision trees, and then the weights are calculated, which helps us know the relations between the different genes. This network is then stored in east_grn.csv.